Welcome to the IACM Cannabis Talk with Dr. Franjo Grotenhermen.
Hello everybody, I would like to welcome for our second Cannabis Talk Daniele Piomelli. He is Distinguished Professor of Anatomy and Neurobiology at the University of California in Irvine. He studied pharmacology and neuroscience at Columbia University in the 1980s with Eric Kandel and the late James Schwartz, and at Rockefeller University with the late Paul Greengard. And two of them, Greengard and Kandel, they received the Nobel Prize for their contribution to medicine and physiology. He had several milestones in his career, the INSERM in Paris. He was also at the University of Naples. And finally, he went to the University of California, where he is now Professor of Anatomy and Neurobiology. And I'm also very happy that he is a long-standing board member of the IACM. He is a very important person in the USA with regard to cannabis medicine. He is director of the University of California in Ivy's Multidisciplinary Center for the Study of Cannabis and directs the NIDA-supported Center of Excellence, ICAL, Impact of Cannabis Across the Lifespan. So we have here a very experienced cannabis researcher, basic researcher, and I'm so happy to talk with him about the endocrine system. And what I can say is that Daniele is not only an excellent researcher and a speaker at conferences, but also very pleasant and good communicator and helping very well with the IACM to keep it going. And so now let's go into the topics. And my first question is, how came you to cannabis and cannabis? It was not so well known in the 1980s, so how did it, why did you come to this field?
Well, thank you, Franjo, for your kind words of introduction and for having me today. You're asking me to go back to the early 1990s, where I became involved in cannabinoid research. And unlike some of other colleagues, I'm sure you will be talking to, like Aline Howlett or Roger Pertwee, but mostly Aline. I was not interested in cannabis at the time. Actually, I have to say this. As a young man, I was against all sort of psychoactive drugs, so I didn't touch any of them, and thought that they were a means of the power that be to control the population. So I was actually very much against that. So my interest in cannabis did not start from my personal experience with it, but rather from a completely different perspective. I was working at the time. I did my work at Columbia University and then at the Rockefeller University, as you mentioned, to studying the properties of this lipid-derived intracellular messenger called arachidonic acid. And I was working in the aplysia system, which is a marine mollusk, and then in the rat and mouse brain. And I spent my entire PhD period and postdoc period working on that. Then in 1990, in December 1990, I was happily working in Paris, France at the time, on arachidonic acid, and Bill Devane's and Rafi Meshulam's paper came out, in which they isolated from the porcine brain, from the pig's brain, anandamide, and showed that anandamide, this compound they isolated, was able to bind to and activate cannabinoid receptors. And actually Roger Pertwee showed in that same paper that purified anandamide was able to produce in vitro the same effects as THC produces. So that paper came out at a time was I was getting a little bit bored to work on arachidonic acid. To tell you the truth, was I'd been my thesis, my postdoc, so I'd done 6-7 years on that. There was a lot, and over the last couple of years, you will find this interesting. Over the last couple of years before Rafi's paper came out, I had, for some reason, become interested in cannabis. But theoretical, it was a platonic interest. I didn't use it or anything, but I was.
You never used it?
No, not at all.
Oh,
never until that point. I'll tell you later. I'll tell you later.
It's okay.
But
it's okay.
At that point, I'd never even. I actually hated the idea because, you see, where I come from in Italy, people don't use marijuana. People use hashish, right? So they mix hashish with tobacco and smoke that. I cannot stand tobacco. It makes me physically sick. So I couldn't even get close to, even if I wanted to, which politically I didn't want to, but even if I wanted to, I couldn't, because I hated the smell of tobacco and I hated the idea of smoking. So even though I had no interest in that, I saw I saw my friends literally. This is exactly what happened. Under the influence, I always wonder what the heck is going on there. Why is it producing such funny effects? And in '88, Aline and Bill Devane's paper came out in JPET, in Journal of Pharmacology Experimental Therapeutics, showing that there was a receptor for THC. And that triggered really my interest, and I really wanted to study the endogenous system that activated this receptor. It felt it was so important, but at the time I thought must have been something like a peptide of sorts, because this is the late '80s, early '90s. Peptides were the rage at the time. Everyone was working on peptides. And, for some reason I thought, well, there is a receptor for cannabis, must be a peptide receptor. I don't work on peptides, so why? Too bad, because it's an interesting topic. And then Rafi's paper came out in Science in December 1990, and 1992. And when the paper came out, there was the structure of this endogenous cannabinoid right there. One of the figures contained the structure of the compound that they called anandamide. Bill Devane called it anandamide. And it was a derivative of arachidonic acid. Not only was it a derivative of arachidonic acid, the molecule that I had worked on for my thesis and my postdoc, but it was a very strange derivative, because at the time, all derivatives known were oxidated products of enzymatic oxygenation, like the prostaglandins, the leukotrienes, the epoxygenase products. These are all arachidonic acid attached to it, an oxygen molecule, an hydroxyl group, or something like that. So that was completely strange, and I remember very well seeing the paper, and I was in Paris, France, and so I ran to the office of the head of the department, Jean-Charles Schwartz, which actually this year is his 80th birthday. And I said, Jean-Charles, we have to work on this. And he was like, yeah, it's amazing. We should work on this. So with his blessing, I started working on the pathway of formation of an anandamide, because it was literally impossible to conceive of a way to produce an anandamide according to what was known about arachidonic acid at the time. So that was how I got interested in that. But just to finish something, I don't want to sound like I'm trying to hide my experiences with cannabis. This is something that a lot of us scientists do, but I abhor that. When I started working on it, after a couple of years, I decided, what the heck, I'm working on this. I don't even know how it feels. And so that's when I decided to test it, and it was a very scientific, of course. No, seriously, I went in with taking notes and stuff. It was very kind of cute. I was trying to sort of self-experience it, and it was really important. I got to say, the one thing that made me decide that was funny. But I was in Paris, I told you. And I love old books, so I was, scavenging always for old books, and they found the famous book by Jean-Jacques Moreau de Tours, who is who first in Europe wrote about cannabis. It's a beautiful book. I found the original edition, and immediately bought it.
The Club of Hashish Eaters, yes?
Come again?
In Paris. It was among the club of hashish eaters in
Oh, there
Paris.
was a club of hashish eaters in Paris at the same time. This is 1845 about, yeah?
Yeah.
But he was part of the club, but he was also a very keen observer. And he describes a series of effects that cannabis has on people that are so very well detailed. He even actually, interestingly enough, he even tried it in people who he diagnosed what something today would be called schizophrenia. The symptoms are akin to those that we would call today schizophrenia. And Jean-Jacques Moreau, he worked at the hospital, the Sainte-Anne Hospital, Hôpital Sainte-Anne in Paris, which is where I was working at the time. I was working just next to the Hôpital Sainte-Anne at the INSERM, Unité Saint-Neuf. And so I felt there was a call, and he, in the introduction, he said, "You cannot talk about this unless you try it." Okay, I said, "Well, if Jacques Moreau de Tours says that, I'm going to do it." So that's when I started trying it, and of course it was illegal, so I didn't do it very often. I did it a couple of times. I had my own experiences, and stopped. But I have to say that when it came back, when it became legal for non-medical use in California, I tried it again because I felt again that it'd been a long time and I wanted to understand better. And I have to say it did help me understand. It did help me understand better the effects and what animals experience when we administer that. You cannot tell, of course, what an animal is experiencing, but if you have your own personal experience, you can at least have an idea of what might be going on. So that's really my story about that.
Okay, and what were people thinking about the action of THC before the detection of the endocannabinoid system?
Oh, it was a jungle. Yeah. It was a jungle, Franjo. It was so interesting because you see the structure of THC, has got a very oil-soluble or hydrophobic component and a couple of little hydrophilic points, like the OH, the phenol group, the OH phenol group. So that means that put people on the wrong direction, because they thought this before the discovery of the receptor in '88. They thought that it would be kind of a soap, because soaps are like that. Soaps have a very big lipophilic or water insoluble component, which is typically a long chain, right? And then they have a head that is a polar head, right? So that's why they form micelles. Micelles have got all the polar heads around, and inside there is the hydrophobic part. So people were thinking, very serious people who are still in the field, exploring the possibility that it could interact with membranes in a non-selective way and cause...
Similar to alcohol or something like that, yes?
Come again?
Similar to alcohol or something like that.
Similar to alcohol.
Unspecific or something?
Non-specific activation. But I never thought that they was credible. And the reason for that is that it seemed so impossible for something so non-specific to produce effects that were so peculiar. I mean, if it had been an anesthetic, like, an inhalant anesthetic, a gaseous anesthetic, I would have understood. But it's not anesthetic. Depending on the dose, it can stimulate, it can produce very peculiar effects like making people laugh. I mean, one of the classical examples of one of the classical signs of cannabis intoxication is everything seems funny, and then you get hungry. Now, these are the good effects, right? The effects that people typically seek out in a cannabis experience, non-medical cannabis experience. So everything looks more interesting and funny, and then you're hungry. So that these are the good effects. There are others.
So there were people who thought that this cannot be real? There must be something specific action,
Yeah,
or what was the
that was
idea?
my thought. That was my thought all along. I had no idea, of course, that there was a cannabinoid receptor. There was the most abundant G-protein coupled receptor. Well, we know that now. I had no idea, and for crying out loud, I had absolutely no idea that there were cannabinoid receptors and endocannabinoid system outside the brain. That seemed to me completely outlandish. I didn't even think about that, and now we know, in fact, that that is the case. That the endocannabinoid receptor is probably as important outside the brain that it is inside the brain.
And what allowed the breakthrough to really detect the receptors? What was necessary to what tool or what idea or what finally led to the detection of specific cannabinoid receptors?
So, as usually in science, it's not a single thing, but two things in this case I think were critical. One was the pharmaceutical industry was interested in cannabis, particularly in THC. As you know, THC had been isolated in the 40s, but until the 1960s, 1964, the structure was known but not completely determined. So in 64, Rafi Meshulam determined the final details of the structure of THC and synthesized it. So at that point, people noticed that it was an analgesic compound. Not a very good analgesic between you and me, but an analgesic compound. So they started to, people in pharmaceutical industry started to pay attention. And what do? The first thing that pharmaceutical scientists do is to make analogs, make compounds, which they can patent and then sell, right? That's the name of the game. So there was a company called Pfizer, which is still around, as you well know, that was interested in this. And chemists at Pfizer were trying to make analogs of THC that were better than THC. And they succeeded very well. They made these super potent compounds. And that was important because among the various things that they did, they were able to also create compounds that were optically active. So these are compounds that have, that can be, that have enantiomer. So they have an R enantiomer, an S enantiomer, but chemical structure that were a little bit simpler than THC. And so with this group of now new molecules, which again had no receptor at the time, no one knew that there was a receptor, Aline Howlett comes in the picture, and with her student, Bill Devane, who is passed away, unfortunately, very young. But Bill, who was a student with Alan, was interested in cannabis for reasons that, unfortunately, I don't know. I spoke to him several times, but I never asked him this question. He was interested in cannabis, and he tried these different analogs in rat brain extracts, rat brain membranes. And what he found, and it took a while because this was not an easy experiment at the time, what he found is that there is a binding site in the rat brain membranes for these various compounds. Again, now the compounds were available, not just THC, so it was possible to rank their potency. So let's say you have a compound that is very potent, like CP55940, which was a Pfizer compound, very potent, so you have a certain dose response.
So this Pfizer compound is binding much more efficiently to the cannabis receptors than THC, which has a rather low affinity.
Right.
And so since it binds more strict, more stronger, then you can see it better. Is it right?
Yeah,
this way?
That's certainly one reason. But you can also, I mean, if you don't know that there is a receptor, how you tell there is a receptor? One of the ways, and I'm sure you can ask Aline about that, and she will give you many more details and be more accurate. But basically, one thing you can do is to say, okay, if this particular compound is very active in vivo, so I give it to an animal at 1 milligram, and the animal goes, completely stoned, and it's, I need 10 milligram of THC to get the same effect. In vitro, there should be the same rank order potency, right? So you should see the same rank order potency. And so of the binding to the receptor, and also what they did in that study, 1988 JPET paper, what they did was they also determined whether this was a G protein coupled receptor, which at the time were very fashionable, was something people were studying a lot, and Aline had studied them before, and she was interested in this, has been interested in this for a long time. And so they were able, using these Pfizer compounds along with THC, to identify what they call the binding site present in membranes that bound to THC and was able to elicit a G protein-mediated response, so a change in GTP, gamma rays, doesn't matter the detail, but that's basically what it was. So that paper, for 2 years, nothing happened. And then in 1990, if I remember correctly, yes, I do remember correctly, by chance, Lisa Matsuda, working in Tom Bonner's lab at the NIH, found the cannabinoid receptors. And let me tell you how I was told by Miles Erkenham, who was at the NIH at the time. He still is, I believe. And it's important in the story for another reason. So Miles Erkenham also was interested became interested in cannabinoid receptors, and he was able to obtain from Pfizer this highly potent cannabinoid receptor agonist, the CP compound. And he obtained it not just as just a regular molecule, but also labeled with a radioactive tag. I think it was tritium, or could be C14, I don't remember now. I will have to look it up, but it was a radioactive tag. So now he has this highly potent agonist, and he wants to ask the question, does it bind to the receptor in a brain slice? So he takes brain slices, fixes them, puts the radioactively labeled compound on top of them, washes out the excess, and puts the slices against a photographic plate and waits for 2 months. 2 months later, it takes the plate off and sees if there is any binding that could be detected and makes biological sense. And lo and behold, there was a lot of binding that made a lot of biological sense. In fact, he saw, this is in the rat, he saw a ton of this binding in the cerebellum. He saw a lot in regions of the brain like the striatum and the medulla, not the medulla, sorry, the substantia nigra that had a lot of cannabinoid receptors, and very nice binding in the cortex, which you would expect it to have because of the cognitive effects of THC. And he did all the right controls. Miles Erkenham is one of the best anatomists that I know of, and he did a terrific job at that. And that paper came out in PNAS in 1990, saying, okay, look, there is a cannabinoid binding site in the rat brain, and it looks like it makes biological sense. At the same time, the same year, a couple of months later, Bill Devane, who had now moved from Alin's lab to Rafi's lab in Israel, in Jerusalem, he comes out with a paper where he isolated this lipid from porcine brain. And there's a funny story about that I need to share with you. He isolated this lipid from porcine brain, an enamide, which bound to the receptor. So it was a double whammy. There was, on one hand, the localization of the receptor in the brain. On the other hand, there was the ligand right there, and it was just a story ready to be, a gigantic story ready to be unraveled. Now, I want to tell you this part of the story that makes it even more interesting. No one believed this at the time. I want to make sure that you know this, because now it's like, oh, the endocannabinoid system. I'm an expert. Everyone is an expert on the endocannabinoid system. There is a job of endocannabidologists, apparently. Well, no one believed in this. No one, I'll tell you, and in part for good reason. So here's something that happened that almost got me lose my job, but eventually turned out to be a good thing. So I published a paper in 1994 showing how an enamide is produced. And next year in JBC, in Journal of Biological Chemistry, a very eminent chemist, lipid biochemist whom I knew fairly well, published a paper stating that there was no anandamide in the rat brain. So I had worked on an anandamide in the rat brain. I'd worked on brain neurons. I knew there was an anandamide, but, analytical techniques, etc., at the time were not perfect. And now this guy, who is, a big star in lipid biochemistry, tells the world that there is no such a thing as an anandamide. I wasn't shocked. Shock is not the right expression, but I was clearly surprised, but mostly because at that point I'd quit France. I was back in the States. I was in California. I had my own mass spec, which I couldn't do in France, and I could see an anandamide. I could see an anandamide. The peaks in it was not an LC mass spec, it was a GC mass spec. That tells you how old I am. But I saw an anandamide. So I called up this guy. His name is John Turk, and they said, John, I don't get it. Why don't you see it? I see it. So maybe I can send you my samples. You can send me your samples. We got to figure this all out, because my employer at the time was kind of a little bit of a cuckoo guy. And let's not mention his name. It was like all angry at me. Oh, what is this? You're cheating. Whatever. Some crazy stuff. The guy is dead now, but he was known for being rather crazy. Anyway, so John Turk and I talked about this, exchanged messages about this, and eventually we arrived to the conclusion that the reason why John had been unable to see an anandamide in the rat brain is because the way he had set up his assay, he could only get really high levels of an anandamide, the levels that Rafi had seen in the pig brain. And they were not there in the rat brain. The rat brain had much less. So John had set up his assay so that he could capture the levels of an anandamide found in the porcine brain, as published by Rafi. And I, because I was an ignorant ass, and I was a kid, playing with something, I did not even pay attention to the quantity. I set up an assay that allowed me to get nanomolar and picomolar levels. So I saw it, but John couldn't see it. So when I shared with him this, he did the experiment and found it and said, okay, so we are all fine on that. So, but I was curious. So I went and asked Rafi. I said, Rafi, how come you have such high levels of anandamide in the pig brain? And he said, I don't know. I said, wait a minute, so you're in Jerusalem. I said, where do you get the pigs? This is, like in Jerusalem, either is kosher or is halal. There are not very many Christians. There are some, but not very many people eating pork in Jerusalem. So where the hell you get your pigs? And he said, I get them from Cyprus. They ship them? Yeah, they ship them from on ice from Cyprus. I said, that's cool, but how long it takes for them to arrive? A day or two. Okay. So one thing you need to know, probably now it's forgotten, but one thing we have learned meantime about anandamide is that if you take a brain and you let it sit on a bench, a brain of a rat or a mouse or a pig, you let it sit at room temperature or not frozen, anandamide levels increase. They skyrocket. Anandamide is produced post-mortem in large amounts, which poses analytical problems, of course, as you can imagine. So that's how Rafi had discovered anandamide. He was able, Rafi and Bill were able to isolate it because they were working on pig brains coming from Cyprus. And these pig brains had enough anandamide that they could extract this lipid out of a whole tissue and run the assays. So it was
fascinating story. Fascinating story.
Very
really
strange things, And I'm so sorry that Rafi is not with us to tell his side of the story, because when he told me that they were coming from Cyprus, it was like a light bulb went in my head. Anyway, so that's a little bit of an anecdote on the side, but I thought it was pretty funny.
Very interesting, fascinating. That's sometimes how science works, huh?
Yeah. And I'll tell you, there is a segue to this. And, I told you that I almost lost my job. I almost did, because I was at the Scripps. I was part in something called the Neuroscience Research Institute, or the Neurosciences Institute rather. And the head of that place was a little bit of a sociopath. Actually, he was a total sociopath. But one thing that he basically thought that I was cheating because if there was no anandamide, what I was working on, and I could show him the LCMS tracings, but he was not that kind of scientist. He was doing something completely different. So I actually had to, and I'm actually quite happy that now of the way these things developed, I had to publish a paper where we characterized not just the basics of anandamide production, but every single step. And for every single step, we had mass spectrometry data completely and solidly identifying the precursors, anandamide, all the other fatty acid ethanolamides, and the enzymes involved. And that paper was published in J Neuroscience. So the first paper, the 94 paper in which we identified the pathway, was published in Nature. But in 96 and 97, we published these two papers in J Neuroscience where we characterized the mechanism. And the 97 paper had so many mass spec tracings there. It was like, I don't think J Neuroscience ever published so many mass spec tracings before or after. And I think he had 12 figures, plus as many tables. So it was a substantial effort. But that saved me because, when we published that paper, no one could doubt anymore, which has been replicated by many, of course. No one could doubt that anandamide was real. But let me tell you, anandamide almost died because of that 95 JBC paper by John Turk. It almost died.
No, very good, very good. When we talk about the endocannabinoid system, our concept or our ideas of what the role of the endocannabinoid system is considerably changed from the end of the 1990s to today. Yeah, there was a considerable evolution. Perhaps we can talk a little bit about this evolution. You can tell me what the endocannabinoid system looked like at the end of the 1990s, and today we sometimes speak of the endocannabidome or something like that. What we think about the endocannabinoid system of today. You told me already that you couldn't imagine that there are cannabis receptors outside the brain, for example. Yeah. Perhaps you can tell me a little bit about the evolution of our concept of the role and the compounds of the endocannabinoid system in the late 1990s and today.
Yeah. So the first thing to appreciate is that nothing was known. When you have in 1990, until the discovery of the receptor and the discovery of anandamide and the characterization of the pathway of anandamide, and then the discovery of 2-AG, the characterization of the pathway of 2-AG, these were important steps, but we knew really nothing about the physiological role of this. And one of my concerns was that, I was a neuroscientist by training, and I knew that the neuroscience community would have had a very major impact if they became interested in the endocannabinoids. But they were not. They were not. And one major reason for that is because there were not enough tools. Okay, there were not, there were no knockout animals. There were not even antagonists at the time. The first antagonist was published in '94. So what I tried to do really, and I don't want to take credit for this, but it is a fact that I was trying to get the scientific community, the neuroscience community, to become interested in cannabinoids, because I felt that because neuroscientists are usually very high quality science, they produce really high quality science, I figured that they would shed light on something really, really important alongside with people who were more interested in kind of, the basic cannabis pharmacology, who were interested in, and had been interested for a while. So I think the thing that changed that, the thing that actually moved the needle seriously toward cannabinoid research becoming a neuroscience thing, and now there are like thousands of papers published on the neuroscience of the endocannabinoid system, was actually the discovery that that 2-AG is produced in the brain, which Nefi Stella in my lab and I did in '97. We had a Nature paper on that, and that it modulated a form of synaptic plasticity called long-term potentiation. So that was a paper that attracted the attention of neuroscientists because long-term potentiation of LTP was a hot topic at the time. So when they saw that there were these molecules that could work in the brain, they started paying attention. And what they discovered actually is something that still remains, is still considered the key property of the endocannabinoid system in the brain. So they discovered that an endocannabinoid could be the mediator of a phenomenon that doesn't matter what is it called, but a phenomenon that of postsynaptic to presynaptic communication. Basically, the famous retrograde messenger role of the endocannabinoids, which has now become almost like a catchphrase, right? And it was in 2001 that, in the same year, 2001, 3 years after the publication of our Nature paper with Nefi Stella showing the production of 2-AG in the hippocampus by appropriate physiological stimuli, and the fact that application of 2-AG would produce inhibition of LTP, that various labs around the world, in the United States and in Japan mostly, 2 labs in the States and 1 in Japan, showed that an endocannabinoid mediated retrograde signaling, which was at the time the super hot ticket that everyone was excited about. So now they became excited, so neuroscientists became excited, and that really changed. I want to say that changed a lot as far as the brain is concerned, right? As far as the brain is concerned, that changed a lot. It turns out actually that it's not any endocannabinoid that mediates retrograde signaling, but rather is one endocannabinoid, 2-AG. Anandamide does not work that way. But now I'm trying to hammer in the head of the neuroscientists that there is a difference between 2-AG and anandamide. Anandamide is not a retrograde messenger. Anandamide is a volume transmitter. Think of it as a volume transmitter. Adenosine, 2-AG is a proper retrograde messenger. And actually, in my lab, my associate, Kwang Muk Jong, who is originally from the Republic of Korea, he did all the work to show that indeed there is a signalosome, so a complex in the postsynaptic spine that is able to release 2-AG when it's activated by glutamate acting on metabotropic glutamate and GLUR5 receptors. And then, we showed that there is this enzyme MGL that is in the presynaptic bouton, and MGL is cytosolic but can bind to the membrane and cleave 2-AG. So building this little model that now is, in the textbooks took a long time and a lot of work. And now, unfortunately, I'm sure that the model is only partially right, but now, of course, it's in the textbook, so everyone thinks it's what it is. But, I think it still needs work. We still need to understand better how this works. So that is one part of the one thing that changed between the 1990s, early 1990s, and the beginning of the 2000s. In the 2000s, there was a sort of this transition point where, it became a big neuroscience thing. The other important thing is that the French company Sanofi was interested in cannabinoids. They discovered the first cannabinoid receptor antagonist, both the CB1 and CB2, and their CB1 receptor antagonist, which now goes by the name of rimonabant, at the time was called SR 1417-16A, and that compound they tried in all sorts of models. They gave it to everyone. The CEO of the company at the time, he was the scientific director. His philosophy was, we're going to give it to the scientists. They're going to do their job. If there is a problem, they'll tell us. And I think it was a very smart idea. His name is Gerard Lefure. He's retired now, but he's a smart guy. So Gerard Lefure, he gave the company away, And me and 100 other different labs used it in a bunch of different things. And one thing that started to appear is that this compound had all these effects on metabolism. Right? So, to make a long story short, Sanofi, which initially was interested in potentially using rimonabant for schizophrenia, decided to test it for obesity. And they tested for obesity, and it worked really beautifully. By beautiful, I mean it was extremely effective. They did a bunch of clinical studies where they found two things that are important. The first is that you have, an improvement of the overall metabolic profile, and people lose weight, their waist size reduces, etc. Substantial dose-dependent effects, so it was good pharmacology. And the other thing that they found is that this was not due to a change in food intake, which is what everyone expected. THC gives you the munchies, you eat more. So if you stop the effects of THC or of an endocannabinoid, you eat less. But they only last for a few days. Yeah, if you give rimonabant, the CB1 antagonist, to people and animals, they will eat less for a few days, but then they recover. They go back to normal eating. So what is causing them to lose weight? Well, that's when Daniele Coda and Uberto Pagotto and others found that there are cannabinoid receptors in adipocytes. And if you activate those receptors, you make adipocytes fatter. If you block the receptor, you make adipocytes thinner. Adipocytes grow and in size depending on how much fat they incorporate. So blockade of the cannabinoid receptor in adipocytes makes them thinner. And that's the effect of rimonabant, not in the brain, not in the hypothalamus. That is an effect that lasts only for a few days. The animals and the humans adapt, but the effect in the adipose tissue does not. And now, fast forward to a few years ago, we know now that the main function of these peripheral endocannabinoids, one of the main functions of the peripheral endocannabinoids, I don't want to be too dramatic on this, but certainly one of the functions that was most unexpected, that's for sure, is that it regulates not just food intake, but also energy balance at large. So energy balance at large means the intake of food, but also the storage of nutrients. So by and large, what the endocannabinoid system does is a thrift system like insulin. It stimulates hunger. It increases the absorption of lipids, of fatty components, and it increases also the storage of those lipid components in our adipose tissue. So it's a thrifty system that was very likely put in place by evolution to allow animals, whenever they have access to food, to eat, probably to fatty foods in particular, to eat as much as possible of it, and store it at the highest possible level of efficiency. Because as we all know, in the wild, it's not like, you go to the supermarket. In the wild, it takes a while to find fat. There aren't very many fat animals hanging out. You need to, our ancestors had to break, this, right? They had to break the bones and suck the marrow, which is one of the fattest part of the body. They would eat liver, they would eat internal organs. That's the prize food because it's a lot of fat. They will eat brains because that contains a lot of fat. Muscle, they would actually probably throw it to the dogs. What we eat now is prize meat. They would probably throw it to the dogs. The endocannabinoid system, and this is, I think, the most unexpected thing, really is important in regulating energy balance. And that is something that was completely unexpected in the 1990s. And now I think it's probably I don't think anyone doubts that. Aside from that, I think the other big difference is that we are still, what would have made a huge difference, it still hasn't, and I'm still hopeful for the future. We don't have an endocannabinoid-based drug. we don't have an endocannabinoid-based drug. So there is THC, of course, which is in the clinics, but THC is not an endocannabinoid-based drug, it's a cannabinoid. An endocannabinoid-based drug would be a drug that either activates or stops the endocannabinoid system. And stopping the endocannabinoid system turned out to be complicated because although it's very effective at reducing body weight and improving metabolic profile, it also has pretty substantial psychological effects. It causes anxiety, depression, suicidal ideation, stuff like that. So cannabinoid receptor antagonists never made it to the clinic. I mean, they made it to the clinic, they never made it to the market. In Europe, they did shortly, then they were withdrawn. What we need now, I think, to make the next step, qualitative step, and we're not there yet, but people are working on it, including my own lab, we are working on it, is drugs that utilize the endocannabinoid system for a therapeutic purpose.
Yes, yes. And you mentioned already that in the first years there was the idea that the endocrine system is an issue of the central nervous system. And you also underlined that you find it in fat cells.
Yeah.
So the detection of the endocrine system in the periphery was an important step. We now find it in bones, in the skin, in everywhere.
Was a major step. Look, if it wasn't for cannabis, we would not know. And this is actually the greatest debt that we have toward the plant. The reason why we know that there is this endogenous cannabinoid system regulates pretty much everything in the body. It regulates it. It doesn't control it. So there is it has a modulatory function, but a modulatory function that is important, clearly, and not just in the brain, is important in the vasculature, as the work of George Kounos has demonstrated, is important in the regulation of peripheral pain processing, as a bunch of people like Andrea Homan in our lab has shown, is important in the regulation of adipose and hepatocyte biology. And actually, George Kounos again did some really fundamental work, particularly on both hepatocytes and adipocytes, to show that blockade of cannabinoid receptors specifically outside the brain is sufficient to produce a therapeutically important effect in patients, in people with obesity. And he characterized all the molecular pathways involved in this. So we know now that the endocannabinoid system is involved in a variety of very important physiological functions. But again, I think, having worked on this for a while, I think that is a hierarchy, and I think that when it comes to the periphery, the main role is the regulation of energy balance and, I want to say, pain next to it. And when it comes to the CNS, the main role is the regulation of mood and the response to stress, and possibly also hunger, but not so much other functions. Although, of course, THC can have effects on other things, like, memory, for example.
Yeah. We talked already about 2-AG and anandamide, but there seem to be more endocannabinoids even in the body. And usually a body does not produce substances just like that, because we know...
That's complicated, Franjo. That's really complicated. Let me try to step back for a second and go there. It's a really important question. Thank you for asking it. So we have to be, I think, scientists tend to be really good with experiments, but not very good with words. That's a problem that we have as a category. So I want to spend some time in saying, what is an endocannabinoid? To me, an endocannabinoid is a molecule. I'm agnostic about the structure, but a molecule that engages the endocannabinoid system, so activates the cannabinoid receptors, CB1 and CB2. So if we use this sort of a restrictive definition of an endocannabinoid, we have two major endocannabinoids, anandamide and 2-AG, and closely related molecules. Anandamide is a derivative of arachidonic acid. Arachidonic acid has 20 carbon atoms and 4 double bonds. 2-AG also is a derivative of arachidonic acid with 20 carbon atoms and 4 double bonds. If you remove one double bond or you add another double bond, which in nature happens all the time, you have molecules that are similar to anandamide and 2-AG that will also activate cannabinoid receptors. Those are present in smaller amounts, but are definitely present and are important regulators of the cannabinoid system. So I consider them as cannabinoids. However, if we are talking about lipids that are similar in structure, or maybe they have some kind of biochemical similarity with the endocannabinoids, but they don't bind to cannabinoid receptors, they don't activate the endocannabinoid system. Calling them endocannabinoid, I think, is a mistake. And why? Because I think it makes things confused to people, including to us scientists. Because, you know, when we start thinking, oh, wow, well, so it's an endocannabinoid, I don't know, palmethylethaноlamide.
Sometimes people speak about endocannabinoid-like substances or something like that, which is confusing.
Sometimes we refer to them as endocannabinoid-like substances, and I've used that term myself, but then I decided it wasn't precise enough. So I introduced a term which goes back to the fact that I like ancient languages, as you probably know, and I like in particular Latin and Greek. I like Greek a lot. It's a very ingenious language. And so I propose the term paracannabinoid to define these molecules that are around the cannabinoids. And my good friend Alex Makrianis, who is Greek, would probably agree with me. Para in Greek means ancient and modern Greek, means surrounding, around, something that is around, right? Like in the word paraphrasis, for example, or paraphernalia. These are all words that indicate something around, okay? So the paracannabinoids are compounds that are similar in structure but not identical to the endocannabinoids that serve, they don't bind to cannabinoid receptors, they bind to different receptors. For example, they can bind to GPR119, which is a G protein coupler receptor, or to the nuclear receptor PPAR alpha, or other nuclear receptors like PPAR delta and PPAR gamma. And these compounds have different functions from the endocannabinoids, but what is interesting is that because they share some chemical and biochemical similarity, and they're also produced, they kind of can work either synergistically, so they can work together with the endocannabinoids, or antagonistically, they can work against the endocannabinoids. Let me give you an example. Palmethylethaноlamide. Palmethylethaноlamide, or PEA, is very similar to anandamide in structure. It has ethanolamine, it has a fatty acid. The fatty acid is not arachidonic acid, it's palmitic acid, so it's shorter and it's flat, doesn't have double bonds, okay? PEA does not bind cannabinoid receptors. In fact, Raffi used it in his science paper with Bill Devane, where he discovered anandamide. He used it as a negative control. Now, it turns out that PEA has its own receptor, which is PPAR alpha. It binds also to other things, but PPAR alpha is the main, it's a nuclear receptor, is the main receptor to which PEA binds. And what it does is similar to anandamide and synergistic. What does it mean? It means that if anandamide does 2 and PEA does 2, when you give them alone, if you give them together, they should give 4, right? 2 plus 2, 4. But that's not what happens. You have 2 plus 2, 8. So 2 plus 2 equals 4 would be an additive effect. 2 plus 2 equals 8 is a synergistic effect. Anandamide and PEA have synergistic effects on the control of pain regulation, which is actually a very big and important area. And we discovered actually that back in 1988. We had a Nature paper on that with my good friend and collaborator at the University of Naples, Antonio Calignano. Now he's retired. Good for him. But PEA now is a big thing in Europe. I'm sure in Germany it is, as well as it is in Italy. And it's becoming a big thing in the States as well, and is used to treat pain. That's a result of that an end result of that study. So PA and anandamide, PA I consider a paracannabinoid, and anandamide is an endocannabinoid, and they work together. But they can also work in opposite ways. So there's this other paracannabinoid, which is oleoylethanolamide, so now oleic acid instead of arachidonic acid. Oleic acid is a little bit longer than palmitic acid and has one double bond. So oleoylethanolamide, if you go and look at what it does in the body, the first thing that we discovered back in 2001 was that it completely curbs appetite. It stops appetite. It causes an elongation of the time between two meals, increases what is called the satiety ratio, so that instead of eating 10, you eat 5. And not because you eat less in each individual meal, but because the meals, instead of being very close to one another, become elongated. So we published that in Nature in 2001, and then we had a paper in 2003, also in Nature, to show that the receptor mediating these effects was PPAR alpha. And we published a bunch of stuff on that. No one gave a damn about it, I gotta tell you. I mean, those papers are widely cited, but for some reason, in the field of appetite regulation, they only believe in peptides. They don't care about lipids. Anyway, eventually, I think it would become an interesting topic for others. But anyway, the point I'm trying to make is that anandamide stimulates food intake and OEA blocks food intake. So now you have two similar compounds, an endocannabinoid and a paracannabinoid. They do the opposite thing. So you see, there is a connection, a functional connection, but it's not obvious. It's not something that you can predict.
Yeah.
And so if we call them an endocannabidome or whatever we call it, we are really making a big bucket, and we're throwing everything in a bucket. Now, that's not critical, because what critical means is that you have a bucket and you divide it into multiple sub-buckets, right? So I am more fond of being critical when it comes to stuff like that than putting everything in one pot.
Yeah, I understand. Very good.
And pot,
paracannabinoid. I like the paracannabinoid. I like this term. Very very good. To come to the last question, to find something to clarify another issue, the difference between 2-AG and anandamide. For example, when does anandamide levels increase? I've learned that, for example, if rats don't eat, the anandamide concentration will increase in the intestine. And perhaps you can elaborate a little bit on the difference between the effects of 2-AG and anandamide, so that we understand that's not always the same or something like that. Because you take it in one phrase and say there are the two endocannabinoids and A2AG and they activate C1 receptors and so on. But what is the difference with regard to pharmacology or something like that?
So that's a wonderful question. So let me give you first the easy answer. The easy answer is, when it comes to pharmacology, anandamide is a partial agonist, which means that it never gets full efficacy. You always stop at a certain fraction of the maximum response. 2-AG is a full agonist, so it will get to maximal effect, and that has been known for years. The other thing that we know is that they're produced through different pathways and they're degraded through different pathways. Fine. When it gets really complicated is when we're trying to answer more broadly the question, the very insightful question you're asking about what is it different that they do. So I'm going to give you my opinion, and now is only educated to the extent that I've been doing this for a long time, but is not fully backed by the data. So my opinion is that 2-AG in the brain is responsible for retrograde signaling. It's produced in the postsynaptic spine. When certain stimuli arrive, it travels back to the presynaptic bouton, it activates the cannabinoid receptor there, and it works typically to stop or to decrease transmitter release. In some cases, occasionally, exceptionally, can also produce the opposite effect, but there are exceptions. So that's 2-AG. So it's kind of a point-to-point retrograde signaling mechanism that is unique, and as far as I can tell, is unique in lipid pharmacology. I don't know of any other lipids that does that. Anandamide is different because anandamide is not produced postsynaptically. The enzyme that makes anandamide is presynaptic. And the great problem that we have right now is that we don't really understand very well how anandamide is produced. But if, based on what we know, it's certainly not produced postsynaptically. And one property of anandamide is that it can actually travel farther away from the site of production than 2-AG can. People have measured how far can 2-AG go, and it's not very far. The reason why it can travel so little is because it will be degraded almost instantaneously. There is MGL, which is monoglyceride lipase, which is this major degradative enzyme, but there's also another degradative enzyme called ABHT6, and there are other lipases that can do the job. Anandamide travels farther away. So whereas 2-AG is a point-to-point regulator of transmission at the synaptic level, what I feel, based on the data that I'm familiar with, is that anandamide travels at a longer distance and can work more as a volume transmitter. And by volume transmitter, I mean something that travels maybe a millimeter away, 2 millimeters away from the site of production, and binds to cannabinoid receptors, not just on the presynaptic bouton, but also maybe on glia, on microglia, on astroglia. So a receptor, so it's more of a broad effect. This is in the brain. This is in the brain. It gets even more complicated outside the brain, because outside the brain, really the hell breaks loose, and we have very little information. So I'm not going to go into any detail there, but you mentioned the intestine, and that's an interesting area. In the intestine, in the enterocyte, which is the cell type that lines the lumen, the intestinal lumen, right? So the intestinal lumen is covered by these cells that are called enterocytes, and enterocytes make anandamide. And they make anandamide when the enterocyte is starved. So if you don't feed an animal, they will start making a ton of anandamide. And we know that anandamide, the work of my postdoc now is a faculty at UC Riverside, Nick DiPatrizio, has shown that anandamide activates cannabinoid receptors in the intestine. And this, the signal travels from the intestine through the vagus nerve, drives to the brain, and stimulates appetite. So it's remarkable, but basically anandamide produced in the gut is able, through this sort of rather baroque process of vagal activation, to signal to the hypothalamus and say, okay, now it's time to eat. There are, of course, other components, cephalic components, but that's an important component. Now, so that is something that is unique to the enterocyte. By the way, the enterocyte also produces OEA, oleoethanolamide, which does the opposite of anandamide, right? But it produces after food arrived to the intestine. So when there is no food, anandamide. When there is food, OEA. When there is no food, anandamide stimulates appetite. When there is food, OEA blocks appetite. I think it's a lovely, very elegant system. And I just wish people were more interested in OEA than they are right now. Well, it's good for me because we are the only ones working on it. But I mean, not the only ones. There are quite a few people, but not very many. But I think there is a lot more to be done. Science is a game of numbers. If only a couple of labs work on it, it never gets solved. You need to have many people working together.
Yes. This is a very good final word. I think we could continue this conversation for easily another 1 or 2 hours, because it's a long journey, long road. It was really a pleasure for me to talk to you, and I hope our viewers and readers they will enjoy the conversation. And thank you very much, Daniele. It was a pleasure.
Thank you so much, Franjo. It's October, so I wish I was there with you with a big mug of beer, the two of us having a nice beer in a beer garden. That would have been nicer than this, but thank you for having me. And at least, I look at me. In October, I can still wear a Hawaii t-shirt.
I must say that this conversation was more interesting than any pint of beer or something like that. It was really a pleasure, and thank you very much for this conversation. Bye bye.
Thank you for having me. Thank you. Bye now.